the receptors of pathogen. So you just stop them from expressing, by which you can stop pathogen infestation. So again, we design guide RNA, we construct, we prepare the construct and then deliver it to the plant. And finally, your plant is produced through tissue culture. And these plants now will have your this target susceptible gene family mutation, because of which now if you infect artificially with powdery mildew, you will see that your plants are resistant to powdery mildew infestation. So this is another approach by which you can do genome editing. Now, coming to abiotic stress tolerance mostly involve like drought, heat, salinity, and also nutrient stress. So because of the change in climate, lots of water scarcity is happening. So because of which you need a drought tolerant, like climate resilient plant. Uh, so the idea is to improve water use efficiency in the plant, or to get heat stress tolerance in the plant. And also you can play with the root system architecture. So you can improve the root architecture, by which you can have more in-depth roots in the soil, so it can hold and also take more water from the soil when there is a scarcity. So also there is salinity, which is a major issue in so this salinity can be again tackled with defined pathway genes present in salinity responsive pathway genes. Mostly these are the receptor genes, which actually helps the salt to come inside the cell. So you stop those receptor genes from functioning. Thereby, you can stop somehow. You can maintain your turgidity, or you can stress the salt stress. You can reduce in your plant. Also, you can improve your photosynthetic efficiency in the plant. Again, I told like several pathways are involved in for your photosynthetic, the whole photosynthesis to happen. This includes number of genes, lots of transcription factors. So you can play with those pathways, and you can actually produce plants which are like having higher photosynthetic activity. You can also play with optimizing grain size and numbers. So again, grain size and numbers are regulated by lots of negative regulatory genes present in a QTL of a plant, I mean a genome. So if you play with those genes, if you downregulate those genes, you can actually increase the grain size. By mutating one particular gene, I can increase the grain size, a short grain from to a long grain. Like this, you can also increase the grain numbers, you can increase the panicle size and everything. Thereby, you can increase the yield of the plant. You can also improve nutritional quality in the plant. Now, we very much need these things in our plant, where we have improved nutritional qualities. With University of Agricultural Sciences, we are working on banana and tomato for like improved nutrient content and shelf life improvement. So these are the aspects where by which you can actually improve different nutrient content in the plant. So again, I will give you an example how this works in real world. So if you see, so protoplast, if I talk about protoplast, is basically a cell without cell wall. So if you remove the cell wall, it becomes a protoplast. So it will have its cell membrane and then all the organelles, whatever is inside. So it's the your protoplast. Now we isolate this protoplast through a standardized process. We can see under microscope how they look like. This is this is coming from your chlorophyll autofluorescence. So this red color is actually because of that. So as I was talking about your right border and left border, so this is a TI plasmid, where it can once you infect it into plant cells, this right border and left border will cut and it will integrate into plant genome. So it actually goes inside the plant cell through pores, and then it integrates into the genome because it has a specific like a recombination sequences. So we design guide RNA, we clone them, and then what we do is we transfect this isolated protoplast using this enzyme complex, like guide RNA and your Cas9 enzyme complex. And then you can actually visualize where your things are localizing. So if you see, this is your nucleus. This is GFP. GFP fluorescence is in the nucleus because it is a nuclear localization signal. So you can see here, like if you just go take it to different steps, and then you see if you merge the structures, like you can see it is localizing in the nucleus. So by this you can understand, because your GFP is actually attached to your Cas9, and your Cas9 has formed a guider with guider and a complex, so everything is now localized in the nucleus. So this is how it looks like under confocal microscopy. And this is just a EGFP. If you normally express an EGFP, it will be expressing everywhere. So you can see it in the cell membrane, and then inside also in the cytoplasm. And these are your autofluorescence, the chlorophyll. So once you get this nuclear localized things and you sort them, we have flow sorters by which we can sort them actually, and the positive one we can use further for multiplying. Now you can actually through protoplast you can generate a whole plant. So this process works very well in some plants. So in rice we can see like protoplast can be regenerated into plant, and you can also end of the day get plants which doesn't require any regulation, because there is no such regulatory things, because we are taking it through protoplast for plant regeneration. So as I have talked about the abiotic stress, cell structure, plant cells, everything. Now it's an developing phase where genome editing actually will play a huge role in coming decades, and so it holds lots of promises. Only issue is it has little bit of off-target effect. So sometimes when you design your guideline, it can bind somewhere else, and off-target effects can happen. Again, there is lots of possible ways by which you can minimize your off-target effects. So also the public acceptance of genome-edited crops are crucial at this stage, because farmers are not very much adapted to the new technologies. They are mostly adapted to breeding technologies and everything they're going and getting through ICR and other institute. So genome editing being a revolutionary tool can actually help in going through this climatic change and everything, and improving your yield end of the day, which is the need of the hour. So the future perspective obviously is like precision breeding, the genome editing, and then you can use synthetic biology, which is like basically bioinformatic based tools using which you can design new pathways and things, and this can be clubbed together for sustainable agricultural practices, thereby getting climate resilient plants. So the conclusion and takeaways, I'll say, like plant genome is the powerhouse. Obviously, it has lots of scopes, and genome editing being the tool of the decade now, one of the major revolutionary tool. It can actually be utilized for sustainable agricultural solution. Only thing is potential challenges involves like public perception and acceptance. That obviously government has to work on it and take it through. So thank you, and if you have any question, I'm open to it.
Specific research going on in flowering plants, because the commercial flowers that are used in markets.
Yeah, so one project we have planned with your horticultural science, University of Horticultural Sciences here on orchid. So like flowering time is very like high in orchid. You have it requires lots of time to flower. So we are trying to reduce the flowering time. So it can you can play with the madbox genes, which are actually the regulator of flowering. So those madbox genes we can actually play with to reduce your flowering time. So also you can play with their texture and color, that also you can play with. So lots of opportunities are there with CRISPR, where you can actually utilize those for improving the color and improving your like flowering time you can reduce. So these things are there. So we are already have a plant project with University of Horticultural Sciences.
Thank you, sir.
Good morning, sir.
Hi, good morning.
I'm Rama from the Department of Chemistry.
Yeah.
I want to know, is there any possibilities to sequester carbon from these types of research?
Sequestration of carbon is little bit of complicated process. because it captures CO2 through enzymes. Like Rubisco is the one of the major enzyme which is present in plant. It's an abundant enzyme present in plant, but the issue with Rubisco is along with carbon fixation, it also does oxygenation. So sometimes it does the reverse process, because of which what happens is instead of capturing carbon, it actually starts capturing CO2 like oxygen. So your enzyme is not very efficient. So now people are working. I was in Oxford doing this kind of research where we actually were working on improving the Rubisco activity so that you can capture more carbon. So sequestration of carbon through plants, that can also be done. Now you have C4 plants which are more efficient, where your preliminary capture is happening in your mesophyll cell, and then everything is done in the bundle sheath cells. So C4 system is much improved system in maize, sorghum, all these plants. So there is lots of opportunities with plant. People are already doing lots of research around the world on how you can sequestration, we do sequestration of carbon. But again, with microbes, there is opportunities. People are again doing with microbes, the soil microbes, that also can help you like doing sequestration of carbon.
Thank you, sir. Thank you.
I think I have a question
Yeah.
here.
So we have a
Thank you very much, sir, for your lucid presentation. I'm Dr. Maheshwari from the Department of Zoology, sir.
Yeah, hi.
Sir, hello, sir. Sir, I would like to take a few clarifications from you regarding the quality improvisations in the leaves in the mulberry plant Morus alba, because that
Mhm.
is the sole plant, the sole feeding plant for silkworms,
Yeah,
Bombyx mori.
yeah.
So, anything to do with the improvisation of quality through genome editing? What is the current status of it in that context, sir?
So this is a nice question. We have a long-term project goal on mulberry. Actually, we are working on different aspect, not directly through genome editing, but that is called RNA spray. That also it works through RNA approach only. I'll not talk about this now. So how genome editing can help is you see mulberry plants are infected by lots of viruses. So major issue with mulberry plant is virus infestation, because of which when mulberry this silkworm feed on the mulberry plant, they actually are infected by the viruses, and lots of wealth loss happens. And Silk Board is now putting in lots of money how we can actually stop this. So again, through genome editing, as I have showed you, like yellow mosaic virus in mung bean, how we have stopped. Similar approach can be utilized to target those specific viruses in the plant, and then by this way you can stop at least the virus infestation in the plant. So mulberry plant silkworm can easily feed on. Again, improving the quality mulberry plant, this is not that area where so much work has still been done, but I think lots of scopes are there. So obviously in coming years, obviously people will take up those things.
Yes, sir. Thank you very much, sir. As we have
Thank
done, we are into research in the lines of feed fortification, feed enhancement. So in that respect, so where we can improve the quality of the cocoon that is getting produced,
Yeah,
as well shortening the life cycle into 2 to 3 days, if it is possible. That can fetch a high yield for the farmers. So
right.
in that line, perhaps after the session gets over, I will request you through the email
Sure,
so that
sure.
you can connect me with those people. It will be a big help for us, support for us to take much in that respect, sir. Thank you very much, sir.
Sure, thank you. Thank you. I
Thank
got a
you.
question through this here. Can we identify specific gene responsible for secondary metabolites through this technique? Yes, you can always identify. CRISPR genome editing is a tool by which you can actually identify function of a gene. So if you knock down one gene, you can actually see the changes in the plant, in the phenotypic expression or biochemical steps. So by this approach, again, if you know certain genes are involved in specific metabolic pathway, so either what you do is you isolate, you go for proteomics where you identify the defined proteome, and from that proteome you can identify the genes actually. And those genes, if you target one by one to silence, if they are not known, then you can actually end up the day identify a gene which actually is responsible for that secondary metabolite production. So there is
Thank you,
lot
sir.
of opportunities. Yeah.
Thank you, sir. Thank